Hi, imagine holding the secret code that writes the story of life itself. Yes, this code is known as the DNA, the genetic material which holds all the information about the life to sustain the life and to carry out various functions of an individual. This connects all the individuals with each other, and this is often called as the master blueprint of life. Now, why it is called as blueprint of life? Because it holds all the information about yourself. Anything about yourself, like what will be your characteristics, what will be your height, what will be the color of your hair, color of your eyes, your likes, dislikes, everything is being coded into this DNA. So it makes protein and performs different tasks. So deoxyribonucleic acid and ribonucleic acids. These are the two major types of genetic material which are found in the living system. DNA, it acts as a genetic material for most of the organisms which are present on the earth. While if you talk about RNA, RNA is a supportive material which helps in protein synthesis. Like it helps whatever information you have on the DNA. So it's a coded language. So you can imagine like we have 26 alphabets in English and we create a lot of scripts. We can express our thoughts. Similarly, here you have four nitrogenous bases in the DNA or RNA, which help us to decode or which holds all the information about ourselves. So DNA has the information and RNA will help expressing it. It helps in the protein synthesis. Protein synthesis. While on the contrary, RNA could be the genetic material for few prokaryotes. Like viruses, we all are aware about the COVID-19 virus, where we have seen the genetic material is RNA. DNA is nothing but a long polymer of deoxyribonucleotides. RNA is the long polymer of ribonucleotides. So as we are looking at this, the monomer over here is nucleotide. How one nucleotide is made up of, we will talk about that in the next slides. The DNA length depends upon how many number of nucleotides have been joined together, right? Further, RNA will act as a genetic material for few viruses. In addition to that, it helps in protein synthesis. I had just mentioned it. This functions as adapter. It functions in some cases, you will observe it acts as a catalytic molecule. So it specifically designed to copy the information which is present on DNA and pass it on in the form of proteins. So we can say it helps in expressing the information which is written on the DNA. By the help of X-ray diffraction studies, Watson and Crick have proposed a model of DNA which is made up of two polypeptide chains joined together with the hydrogen bonding. And if you twist this, like imagine a staircase which has different steps together, and suppose you have this two strands here and you have steps. Now you twist this. So how will it look like? That structure will look like this. So you have a beautiful DNA double helix, which is made up of two polypeptide chain, polynucleotide chains, which are joined together by the hydrogen bonds. These hydrogen bonds are formed between two nitrogenous bases. So we'll look at the structure now, the molecular structure now. So basic unit of DNA and RNA is a nucleotide, which is made up of three units. One is nitrogenous base. Second one is your pentose sugar. Pentose sugar means it has five carbons. So five carbon sugar, which could be ribose or deoxyribose, depending upon whether it is RNA molecule or a DNA molecule. And the third part of it is a phosphate group. So it is made up of three molecules. you have two major types of nitrogenous bases, purines, pyrimidines. Purines are adenine and guanine, while pyrimidines are cytosine, thymine, and uracil. Now, earlier I had mentioned that we have four alphabets, which are nitrogenous bases, but here you will observe that in case of DNA, you will have four nitrogenous bases, which are adenine, guanine, cytosine, and thymine. While if you talk about RNA, there are again four nitrogenous bases, which are adenine, guanine, cytosine, and instead of thymine, you will have uracil here, right? So that's the difference. So there are two major differences that we have observed so far in DNA and RNA, that RNA will have ribose sugar, while your DNA will have deoxyribose sugar. And second difference is in terms of nitrogenous bases, your DNA has thymine. Instead of thymine, RNA will have uracil. So these two major differences. When the nucleotides, they join together, suppose this is one nucleotide, when it joins to the another nucleotide, then another nucleotide. So the bonds which have been formed here are called as phosphodiester bonds. Phosphodiester bonds, and the bonds which have been formed between the sugar and the nitrogenous bases are called as N-glycosidic linkage. So we will understand these bonds further, and we will also understand the structure of a nucleotide, how one nucleotide is formed, right? So we understood the bonds which will be formed between the sugar and nitrogenous base. We have seen that which bond will connect two nucleotides. Suppose this is one nucleotide, this is second, this is third. So this is a phosphodiester bond. This bond I'm talking about, this is a phosphodiester linkage, while this bond which is present between sugar and nitrogenous base is the N-glycosidic linkage. So we'll see further how they form. And when many nucleotides, they join together like four, then five. So when it makes a chain, this chain is called as polynucleotide chain. Now you don't have to memorize these terms, just try to decode the words like nucleotide chain. As many nucleotides, repeated units of nucleotides are there, and poly means many, so many nucleotides, when they join together, they form a polynucleotide chain, and that is nothing but a part of your DNA. Now we'll understand the numbering as well, that what is 5 prime, what is 3 prime. First, before that, we will talk about the nucleotide. So what is a nucleotide? So as you can see here, it has three parts. As I had mentioned, simplified form of nucleotide. You have a sugar, you have a base, and then a phosphate group is hanging over here, right? Now, when I say this is a 5-carbon sugar, that means you have 5 carbons over here. So you have one oxygen here, then you have carbons, carbons, carbons at the edges, and here you have one more carbon, isn't it? So now when you have got 5 carbons in the 5-carbon sugar, now you can give the names, numbers on the basis of this. So 1, 2, 3, 4, and the fifth one. The fifth carbon will be the one which is hanging outside. So let's look at the structure now. So this is your structure of one nucleotide which has a sugar. At carbon number 1, you have a nitrogenous base. It could be, this nitrogenous base could be either adenine, guanine, cytosine, thymine, uracil. On the basis of that, you will have the name adenosine, guanosine, cytosine, thymidine, uridine. So you just change the names. So we'll work on the nomenclature of these later on. Then you have this pentose sugar, which has 5 carbon, 1, 2, 3, 4, and 5 carbons. At carbon number 5, you have a phosphate. So that is how you give the name. So this one is called as the 5' phosphate end of the nucleotide. And this one, as you can see at the carbon number 3, what do you have? You have a OH group. So this is called as 3' OH group. Simple, right? So these numbers are nothing but the number of carbons. Why do you give the numbers to the carbons? Why? Why do we have house numbers? In order to identify where do you live, what is your house number. So that gives your identity, that gives your address. Similarly here, if we have to talk about at which carbon you have base, at which carbon you have phosphate, at which carbon you have OH group. So that is why we have given the numbers to the carbons so that we can identify them easily. So that forms a backbone. Now the next nucleotide will join here, then next nucleotide. So when the bond forms, that forms between 3' of one carbon and the 5' carbon where you have phosphate group. So this forms a backbone. You have sugar, then phosphate, then sugar, then phosphate. So similarly, it will form a chain. So sugar phosphate, that means 3', 5' linkage will be formed in order to make the DNA structure. So you will have two chains like this, two polypeptide chains, and which will twist together to form such helical structure, and these nitrogenous bases at the center will be joined with the weak hydrogen bonds. So we will talk about their functions further. So functions are majorly having information about any individual and passing on to the next generation. So heredity, storing information about the protein. So whatever characteristics we have, like the color of our hair, color of our eyes, our IQ level, our likes, dislikes, everything is being coded into the DNA. That is expressed in terms of proteins. So that's it for today. Bye. See you again.